So, good day everyone. For today's topic is all about the metabolism of carbohydrates. The molecule glucose is the focal point of carbohydrate metabolism, which is commonly called as the blood sugar. Glucose is supplied to the body via the circulatory system, and after being absorbed by a cell, it can either be oxidized to yield energy or stored as glycogen for future use. So we have here a lot of topics on the metabolism of carbohydrates, which will be reported by several persons in my group, and I am Allysa, Pollah, Laccay. I'll be discussing the digestion and absorption of carbohydrates. First up, what is digestion? Of course, digestion, it is the biochemical process by which food molecules through hydrolysis are broken down into smaller or simpler chemical units that can be used by the cells for its metabolic needs. We all know that digestion is the first stage in the process of food products, and the major source of carbohydrates is found in plants. Here is the overview in the digestion of carbohydrates. First up is the mouth, then the stomach, next is the pancreas, and the intestine. The mouth contains an enzyme called salivary alpha amylase. The stomach does not contain any enzyme, and the reason for that will be discussed further as we go on through this topic. Next is the pancreas, which contains pancreatic alpha amylase, and then the intestine, which has dextrinase, maltase, isomaltase, sucrase, and lactase. Digestion in mouth. The digestion of carbohydrates begin in mouth, where the enzyme salivary alpha amylase catalyzes the hydrolysis of alpha-glycosidic linkages in starch from plants and glycogen from meats to produce smaller polysaccharides in the disaccharide maltose. Only a small amount of carbohydrate digestion occurs in the mouth because the food is swallowed so quickly. Although the food mass remains longer in the stomach, very little further carbohydrate digestions occur in there. Why? Because salivary alpha amylase is inactivated by the acidic environment of the stomach, on which the stomach's own secretion do not contain any carbohydrate digesting enzymes. So, obviously speaking, the digestion of carbohydrates temporarily stops in the stomach. The primary site for carbohydrate digestion happens within the intestine, specifically in the small intestine, where alpha amylase this time is secreted by the pancreas, on which it begins again to function. The final step in carbohydrate digestion occurs in the outer membranes of intestinal mucosal cells, where the enzyme that converts disaccharides to monosaccharides are located. There are two phases of intestinal digestion. Number one, it is due to pancreatic amylase, and number two, it is due to intestinal brush border enzyme. Here we have a diagram on the digestion of carbohydrates, the dietary carbohydrates, polysaccharides, disaccharides, and the monosaccharides, down to its product, the glucose, fructose, and galactose. Why cellulose is not digested by humans? Why is it? First of all, cellulose is a polysaccharide found in plants that contain beta-1,2,4 glycosidic bond in its structure. Humans cannot synthesize that enzyme which can break the beta-glycosidic bond. Although it is not digested, cellulose is one of the most important components in the diet because undigested cellulose provides bulk of fiber in the diet that helps in intestinal motility and as a stool stuffer. It softens the stool. Of course, after digestion, we have now absorption of carbohydrates. There are three end products of carbohydrate digestion, and I mentioned that earlier. Number one, glucose, the fructose, and of course, the galactose. These readily absorb into the bloodstream through the intestinal wall. There are two mechanisms responsible for sugar absorption. Number one is the facilitated transport, which depends on the concentration of sugar across the intestinal lumen and mucosal cells. Number two is the active transport, which is an energy-requiring process that requires transport protein and presence of sodium ions. After the absorption into the bloodstream, monosaccharides are transported to the liver where fructose and galactose are rapidly converted into compounds that are metabolized by the same pathway as glucose. Thus, the central focus of carbohydrate metabolism is the pathway by which glucose is further processed. This pathway is called the glycolysis or the glycolysis, which is a series of 10 reactions that involves different enzymes. Next one we have glycolysis or glycolysis. First is we have to define what is it. It is referred to as the Emden-Meyerhof-Parnas pathway, which each glucose molecule is split and converted into 2 or 3 carbon units known as the pyruvate. It is also an anaerobic process which have been necessary in the oxygen-poor atmosphere of prokaryotic Earth. The reactions of glycolytic pathway is divided into two. First is the 6-carbon stage of glycolysis that involves steps 1, 2, and 3. And the second one is the 3-carbon stage of glycolysis, which involves steps 4 to 10. First one is the synthesis of glucose 6-phosphate, also known as the phosphorylation of glucose, to yield glucose 6-phosphate. A glucose molecule with a phosphate group attached to hydroxyl oxygen on carbon 6. The carbon 6 is the carbon atom outside the ring. The phosphate group is from an ATP molecule hexokinase, an enzyme that requires magnesium ion for its activity to catalyze the reaction. This reaction requires energy which is provided by the breakdown of ATP molecule. Next is the conversion of glucose 6-phosphate to fructose 6-phosphate. The conversion of glucose 6-phosphate to fructose 6-phosphate known as isomerization. The glucose 6-phosphate is isomerized to produce fructose 6-phosphate by phosphoglucoisomerase. Here it is, the phosphoglucoisomerase. The net result of this change is that carbon 1 of glucose is no longer part of the ring structure. Next, number 3. The last step in the 6-carbon stage, the phosphorylation of fructose 6-phosphate, known as the phosphorylation reaction that requires the expenditure of energy, in which the enzyme that is involved is the PFK1 or known as the phosphofructokinase, another enzyme that requires magnesium ion. Next, we have the step 4, the first step in the 3-carbon stage of glycolysis, the cleavage of fructose 1,6-biphosphate. Okay, formation of 2 triose phosphates. In this step, the reacting C6 species is split into 2 C3 or triose species. Because fructose 1 and 6-biphosphate, the molecule being split is unsymmetrical, meaning the 2 trioses produced are not identical. One product is the dihydroxyacetone phosphate and the second product is the glyceraldehyde 3-phosphate. Aldolase is the enzyme that catalyzes the reaction. Number 5 or step 5 is the interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Isomerization comes in the formation of glyceraldehyde 3-phosphate. Only one of the 2 trioses produced in step 4, which is the glyceraldehyde 3-phosphate, is a glycolysis intermediate, and the other one, which is the dihydroxyacetone phosphate, can however be readily converted into glyceraldehyde 3-phosphate, meaning the dihydroxyacetone phosphate is a ketose and a glyceraldehyde 3-phosphate is an aldose, which they are an isomers. And the isomerization process from ketose to aldose is catalyzed by the enzyme called triose phosphate isomerase. Okay. Number 6 or step 6 is the oxidation of glyceraldehyde 3-phosphate. Oxidation and phosphorylation. Formation of 1,3-biphosphoglycerate. In a reaction catalyzed by glyceraldehyde 3-phosphate dehydrogenase, a phosphate group is added to glyceraldehyde 3-phosphate to produce 1,3-biphosphoglycerate. The hydrogen of the aldehyde group becomes part of the NADH. Number 7 or step 7. Phosphoryl group transfer. Phosphorylation of ADP. Formation of 3-phosphoglycerate. In this step, the diphosphate species thus formed is converted back to a monophosphate species. This is an ATP producing step in which the carbon 1-phosphate group of 1,3-biphosphoglycerate, the higher energy phosphate is transferred to an ADP molecule to form as an ATP. ADP to ATP. The enzyme involved is the phosphoglycerate kinase, which has magnesium 2. Step 8 is the interconversion of 3-phosphoglycerate and 2-phosphoglycerate. What comes in is the isomerization, formation of 2-phosphoglycerate. In this isomerization step, the phosphate group of 3-phosphoglycerate is moved from carbon 3 to carbon 2. The enzyme phosphoglyceromutase catalyzes the exchange of the phosphate group between the two carbons. Here it is, the illustration. Next, step 9, the dehydration of 2-phosphoglycerate. Formation of phosphoenolpyruvate. This is an alcohol dehydration reaction that proceeds with the enzyme enolase, another magnesium-requiring enzyme. The result is another compound containing a higher energy phosphate group. The phosphate group is attached to a carbon atom that is involved in a carbon-carbon double bond. And last step is the synthesis of pyruvate, the phosphorylation of ADP, the formation of pyruvate. In this step, substrate level phosphorylation again occurs. Phosphoenolpyruvate transfers its high energy phosphate group to an ADP molecule to produce ATP and pyruvate. Here's the ADP and here's the pyruvate in keto form and in enol form. The enzyme involved in pyruvate kinase requires both magnesium 2 and ATP. Again, because two C3 molecules are acting, two ATP molecules are produced. ATP molecules are involved in steps 1, 3, 7, and 10 of glycolysis. Considering these steps collectively shows that there is a net gain of 2 ATP molecules for every glucose molecule converted into 2 pyruvates. Next up is the key points. During glycolysis or glycolysis, glucose is converted into 2 molecules of pyruvate. A small amount of energy is captured in the 2 molecules each of ATP and NADH. In anaerobic organisms, pyruvate is converted to waste products in a process called fermentation. In the presence of oxygen, the cells of aerobic organisms convert pyruvate into carbon dioxide and water. Next, we have the regulation of glycolysis or glycolysis. The control points are the step 1, step 3, and step 10. Step 1. The conversion of glucose to glucose 6-phosphate involves in the enzyme hexokinase. This particular enzyme is inhibited by glucose 6-phosphate, the substance produced by its action. Next, the step 3, where fructose 6-phosphate is converted to fructose 1,6-biphosphate by the enzyme phosphofructokinase. A high concentrations of ATP and citrate inhibit enzyme activity. A high ATP concentration, which is characteristic of a state of low energy consumption, thus stops glycolysis at the fructose 6-phosphate stage. This stoppage also causes increase in glucose 6-phosphate that stores because glucose 6-phosphate is in equilibrium with fructose 6-phosphate. The third control point involves the last step of glycolysis is the conversion of phosphoenol pyruvate to pyruvate, the involvement of pyruvate kinase. The enzyme needed at this point is inhibited by high ATP concentrations. Both pyruvate kinase and phosphofructokinase are allosteric enzymes.